Currently, I am studying second BSc Microbiology in St. Mary's College Autonomous, Thoothukudi. Today, I am going to explain about transposons and IS elements. Hello everyone, in this video we will talk about transposable element which are DNA sequence that can move from one location in the genome to another, and that's why they are known as jumping gene. The binary work on transposons come from Barbara McClintock, and for her work she was recognized and awarded the Nobel Prize in 1983. Let us look at how transposons work. How transposons work can work in two different operational modes. In first mode, they can jump from one location in the genome to the other location, and in the process, they don't leave any copy behind. This is known as non-replicative mode of transposition. In the second, they can make a copy of themselves, and that copy can jump from one location to another. This is known as replicative mode of transposition. So, we can understand that there are two modes of transposition. First is without duplication and second is with duplication. You can imagine that these things to be a cut and paste and copy and paste mechanism. I know you can relate that. Interesting fact is transposable element composes 50% of our genome, but they are generally epigenetically silent. Now the question is, what are the functions of these transposable elements? How they are relevant for us or physiology? It turns out that transposable elements are involved in several diseases like myocardial infarction, neurodevelopmental disorder, neurodegenerative disease, and even it has immune modulatory fractions. That means transposable elements are fairly important, but the question is, do they work? So let's try to understand the part in a moment. Now the jumping gene can jump from one location to another, but the biggest question is, what's the deal about it? Yes, there is a big deal behind this jumping process. Let's say a transposon jumps from one location in the gene and lands inside a coding sequence of a structural gene. That means it would disturb that particular gene. Right now this particular gene has an open reading frame, and this open reading frame is now distributed due to integration of transposon. That means possibly the function product of that gene would not be obtained. There could be a consequence of this transposition. Let's say a transposon hops and land into a regulatory sequence inside a genome. Now this regulatory sequence control the transcription right. So obviously after integration into a regulatory sequence, it can positively or let's say negatively regulate gene expression from a nearby gene. So obviously transposons has activity on gene expression as well as. Let's summarize this part. Transposons can make mutagenic effect. They can get inserted into a location and distribute a gene. They could silence nearby gene by attracting epigenetic modifiers. Transposons can also act as a source of non-coding RNA, and thereby they have great regulatory roles. And lastly, they can act as enhancer or repressor and thereby control gene expressions as well as. Now let's talk about the classification. Different classes of transposons. Of transposons, at least the overview of it, transposons can be divided into three broad classes: DNA transposons, virus like retrotransposons, and retrotransposons. Each of them has their distinct features. Let's talk about the DNA transposons first, which has a particular DNA transposon region known as transposase coding region, which codes for the transposase enzyme. Now, apart from the other feature is the terminal inverter repeats, which is recognized by the transposase. So let's see how they work. So the transposase enzyme recognizes the inverter repeats in the terminal, and they cleave the DNA and creates a double-stranded DNA break. Now this transposable element can be integrated to a new region in the genome, and thereby it can jump from one location to another. So this is known as the integration works. Now let's talk about the virus like retrotransposons. This has inverted terminal repeated as usual, but at the same time it has two genes encoding for reverse transcriptase and integrase. and both of these are important for function of virus like retrotransposons. Let us try to understand that how virus like retrotransposons gets integrated in the genome. So let's try to understand how virus like retrotransposons get integrated into the genome. So first the transposons would be described and reverse transcriptase would make a copy of the and forming a cDNA like structure. So this cDNA or the cDNA intermediate would be integrated into a new location with the help of the enzyme in degrees, and thereby the virus like retrotransposase get into a new genomic location. Let's talk about poly A transposons, which exactly look like a gene. Why is so? Let me tell you. So these retrotransposons differ from other class of transposons but not having the inverted terminal repeats. They have 5' and 3'. Poly A transposons look like a gene, just like our mRNAs. They also have two uh, FRFS and F1 and 2. Lastly, they have but rich region, which the polytail of an mRNA, so they pretty much look like a gene. Now, the F1 encodes for a RNA binding protein and F2 encodes for a protein which has endonuclease and reverse transcriptase activity. Now, exactly how a poly A like retrotransposase works, it's beyond the scope of this overview video is a different video. I will discuss how these particular transposons works. Now let us try to understand more application of these transposers. Are really important in the field of molecular biology, especially the recombinant DNA technology. Let's say you want to create a stable transgenic cell line, so obviously your transgenic construct need to be integrated into the genome, and that it is done by back vectors, which are actually transpose based vector. So this back vector along with the transposase enzyme is injected into the cell, and the cargo is integrated with the help of transposition mechanism, and this integration is fairly stable. So obviously let's say this cargo is a GFP, so you can create a cell line which is stable expressing GFP. Principles of transposition used in ATAC sequencing, which helps research to study chromatin stability. Now, principle of transposition is nowadays used in ATAC sequencing or attack sequence, and this particular sequence technique is used to study chromatic accessibility. So, especially in terms of molecular biology, the principle of transposition is fairly important. Now, let us quickly summarize what we have learned so far. We have learned what are transposons, how do they work, why transposons are important there, implication in diseases and biomedical sciences, and lastly we also learned about different classes of transposition. Transposons. So this is a overview video. Thank you.